Welcome to the next lecture on human genetics and biotechnology, focusing on the human genome. I'm Wendy Nofke, and I'll be giving your presentation today. The human genome includes all of the DNA in the human species. There's about 3 billion base pairs. This includes 23 pairs of chromosomes. In 2003, the human genome was completely sequenced as a result of the Human Genome Project. With our chromosomes, they fall into a couple of different categories. The autosomes are the chromosomes with genes that are unrelated to sex. We have 22 pairs of these. The sex chromosomes are the 23rd chromosome pair. These are going to determine the sex. Two X's will end up as a female, where an X and a Y ends up as a male. In the female, one of those X chromosomes is inactivated as the bar body. That way, the female actually only is expressing one of the X chromosomes at a time as well. The Y chromosome is much smaller than the X chromosome. This is an example of a karyotype here where you can see the chromosomes being paired off. These two here that are significantly different in size are going to represent the X and Y chromosome. The X chromosome has approximately 2,000 genes on it. The Y chromosome is much smaller. It has less than 100 genes on it. None of them on the Y chromosome are essential for survival of the human species. No females have a Y chromosome, so there is nothing on there that is needed for the species to live. What the Y chromosome does have is the SRY gene, which is the sex-determining region of Y. This is going to trigger the embryo to develop into a male. Without the SRY gene being present, by default, the embryo will develop into a female. If for some reason there is a Y chromosome and the SRY gene is damaged or inactive, that individual will by default still develop into a female without the SRY gene to trigger the male development. Only a small portion of the DNA is actually genes. Some of it is regulatory elements, and there's still a very significant portion of it that is still not completely understood. When we look at the number of genes that humans have, we have far fewer than some species who are much simpler. What we are able to do is use splicing to make multiple proteins from a single gene. So we have a more complicated way of managing our genes than the simpler organisms. Most human genes have at least 2 possible alleles. This is going to give more genetic variation. With linked genes, these are located on the same chromosome. Since the chromosomes will tend to segregate together during meiosis, if the genes are on the same chromosome, they'll usually end up both of the genes in question end up going to the same daughter cell, unless they are separated by a crossing over event. With a crossing over event, you have regions of genes or the regions of the DNA that will actually trade places amongst the homologous chromosomes. The closer the genes are together, the more likely they are to segregate together. And this explains why certain characteristics do have a tendency to be inherited together. With the sex-linked genes, these are located on the sex chromosome. Most of them are X-linked just because there are relatively few Y-linked genes on the smaller Y chromosome. These pictures here illustrate the inheritance patterns of the X-linked dominant trait when it's affected from the mother on who it is passed to, when you have the X-linked dominant trait when it's inherited from the father. Here you can see an X-linked dominant trait will only go to female children because those are the ones that will receive the X chromosome from the father. If the X-linked dominant trait is present on the mother, it can go to the male or the female child. The genes on separate chromosomes have a 50-50 chance of being inherited together due to independent assortment of chromosomes. Each chromosome has a 50-50 chance of ending up in either daughter cell. If the genes occur together, more than 50% of the time they are assumed to be linked. The closer they are on the chromosome, the less likely they will be separated by a crossing over event, and the greater percentage of time they will be link together on the same chromosome and show up in the same individual. This information can be used to create linkage maps that shows the locations of genes on the chromosomes. This is created using the frequencies of crossing over that occurs.